For most of the modern era, India and Nepal have been treated as close but distinctly separate genetic worlds. India, the vast subcontinental power to the south, and Nepal, the small Himalayan kingdom wedged between India and Tibet. Its population popularly imagined as some blend of Indian plains people and Tibetan mountain dwellers, sitting quietly in between two much larger neighbors. The two countries share an open border, unrestricted by visa requirements for their citizens, deep religious ties through Hinduism and Buddhism, and a documented history of migration flowing in both directions for centuries. Given all of that, most people assume the genetic picture must be reasonably simple. Shared ancestry in the south, Tibetan-influenced ancestry in the north, and a gradual, predictable blend running between the two. Few international borders anywhere in the world function quite like the one separating India and Nepal. Citizens of both countries can cross freely without passports or visas, work legally in either nation, and in many border districts, families have maintained marriage and trade relationships across the frontier for generations, treating the international boundary as little more than a formality. If any two neighboring populations in Asia were going to show deep, uncomplicated genetic continuity across their shared border, India and Nepal would seem like the most obvious candidates. Scientists decided to test that assumption directly. Researchers compared genetic data spanning both nations, examining Nepal's own extraordinary ethnic diversity. More than 125 distinct groups, from the Indo-Aryan, Brahmin, and Chetri communities of the Terai Plains to the high-altitude Sherpa of the Everest region, against the broader Indo-Aryan and Indo-European genetic framework that defines most of northern India. The question was specific. Given the open border, the shared religious history, and centuries of documented cross-border movement, exactly how genetically connected are these two neighboring populations? And does that connection actually follow the simple north-south gradient most people assume? The answer researchers found was considerably more precise, and in several respects more surprising, than the simple blend zone story anyone might have expected. Certain Nepali populations turned out to be genetically almost indistinguishable from specific Indian populations across the open border. Others turned out to be so genetically distant from India that they share closer ancestry with populations in Tibet and even further east, in Myanmar and southern China, than with any Indian population at all. And one specific Nepali ethnic group, sitting at the historic crossroads of this entire relationship, revealed a genetic signature so evenly mixed between Indian and Tibetan ancestry that researchers describe it as one of the most genuinely admixed populations found anywhere in South Asia. This is the story of what scientists found when they finally compared DNA across India and Nepal. To understand why this comparison produces such a genuinely varied result, you first need to understand the specific genetic framework researchers use to describe South Asian ancestry more broadly. Since 2009, population geneticists have relied on a foundational model describing nearly every population across India as some blend of two ancient ancestral components. Ancestral North Indian ancestry, connected to migrations from Iran, the Caucasus, and Central Asia, and ancestral South Indian ancestry, representing deep, indigenous South Asian roots stretching back tens of thousands of years. This framework describes the vast majority of India's population with real precision, from Punjab in the northwest to Tamil Nadu in the deep south. Nepal's Indo-Aryan populations, predominantly Brahmin and Chetri communities, concentrated across the Terai lowlands and lower hill regions bordering India directly, fit comfortably within this exact same framework, carrying roughly 93% ancestral North Indian ancestry, according to detailed genomic analysis conducted in collaboration with Nepal's own Tribhuvan University. That figure places Nepali Brahmin and Chetri populations genetically closer to northern Indian populations across the border than to many of their own fellow Nepali citizens living in the hill regions just a short distance further north. A Brahmin family in the Nepali Terai, in other words, carries an ancestral genetic profile virtually indistinguishable from a Brahmin family living across the border in Bihar or Uttar Pradesh. The open border between the two countries running directly through what is, at the genetic level, essentially one continuous population. Y chromosome research adds an independent line of confirmation to this same finding. Paternal lineage, studies of Nepali Indo-Aryan populations, have found substantial representation of haplogroup R1a, the same Bronze Age step-related marker found at high frequencies among Punjabi, Rajasthani, and Brahmin populations across northern India. The identical genetic signature this channels earlier coverage of Punjabi ancestry traced back to migrations arriving from Central Asia roughly 4,000 years ago. Finding this same specific paternal lineage at broadly comparable frequencies on both sides of the India-Nepal border reinforces the autosomal DNA findings through an entirely separate genetic marker system, giving researchers considerably more confidence that the Terai's close connection to northern India reflects genuine, deep, shared ancestry, rather than a statistical artifact of any single analytical method. This finding alone confirms something the open border policy between India and Nepal has always implicitly assumed, that populations along this specific stretch of the frontier were never meaningfully divided by the modern international boundary at all. But the moment researchers moved their comparison northward, away from the Terai and into Nepal's hill and mountain regions, the genetic picture shifted dramatically, revealing populations that share almost nothing in common with India's own Indo-Aryan genetic profile. Sherpa populations, concentrated in the high valleys near Mount Everest, carry an extraordinary 94, 3% East Asian ancestry, among the highest concentrations of Tibetan-related genetic heritage found anywhere outside Tibet itself, and about as far genetically from mainstream Indian populations as any group in South Asia gets. Rai populations, according to separate detailed genomic analysis, carry roughly 92% Tibetan ancestry. Magar populations show approximately 82% in some studied groups, and Tamang populations approximately 79%. These are not populations sitting at some intermediate point along a gradual India to Tibet gradient. They are populations whose genetic ancestry connects overwhelmingly to Tibet and the broader East Asian world, showing only a small fraction of the ancestral North Indian signal that defines the Nepali Terai just a few hundred kilometers to the south. This stark internal divide within Nepal itself, Indo-Aryan populations in the lowlands nearly identical to northern India, Tibetan-related populations in the highlands sharing almost nothing with India genetically, explains why any simple statement comparing India and Nepal, as if they were two uniform national populations, misses the real story entirely. The real comparison isn't India versus Nepal. It's India versus specific Nepali regions and communities, each of which tells an entirely different story about its genetic relationship to its southern neighbor. The most genuinely surprising finding in this entire comparison, however, comes from a population that occupies neither extreme, the Newar of the Kathmandu Valley. Detailed genomic research identifies the Newar as the most genetically admixed ethnic group in the entire country, carrying close to equal proportions of Tibetan and ancestral North Indian ancestry, not because the Kathmandu Valley happens to sit at some precise geographic midpoint between India and Tibet, but because the Newar represent Nepal's oldest and most historically significant urban trading civilization, a population whose ancestors, for well over 1,000 years, sat at the literal crossroads of one of the most important trans-Himalayan trade routes connecting Tibet to the Indian subcontinent. The Newar genetic profile doesn't reflect passive geographic positioning between two larger neighbors. It reflects active historical function, centuries spent as merchants, artisans, and intermediaries, directly facilitating trade and cultural exchange between India and Tibet, absorbing genetic contact from both directions, specifically because of the sustained direct human contact that role required, generation after generation. Here is where the comparison develops a genuinely unexpected twist, one that directly challenges the simple assumption most people carry about how Nepal's Tibetan-related ancestry actually arrived, and by extension, how India and Nepal's populations actually relate to one another. A detailed mitochondrial DNA study analyzing maternal lineages across nearly 1,000 Nepali individuals found that low-altitude Nepalese populations carrying substantial East Asian ancestry, specifically Newar and Magar communities, derived that ancestry through an entirely different migratory pathway than the one most people would assume. Rather than descending directly from Tibetan populations, crossing the high Himalayan passes into Nepal from the north, researchers found that these low-altitude communities' East Asian genetic heritage traces back to low-altitude Tibeto-Burman populations who migrated westward along a completely different route entirely, moving from East Asia across the eastern Himalayas, through territory now spanning eastern Nepal, northeastern India, Bhutan, and northern Myanmar, essentially bypassing the high Himalayan passes altogether and entering Nepal from the east and southeast, rather than descending directly southward from Tibet through the mountains. This finding carries a genuinely significant implication for the India-Nepal comparison specifically. It means that some of Nepal's East Asian-related ancestry actually arrived by passing directly through Indian territory, specifically India's own northeastern states, home to substantial Tibeto-Burman populations in their own right, rather than arriving from Tibet independently of any Indian connection at all. In other words, the genetic relationship between India and Nepal isn't simply a story of the Indo-Aryan Terai connecting to northern India, while the Tibetan-influenced hills connect separately to Tibet. Part of Nepal's own Tibetan-related ancestry is itself connected to India, having moved through Indian territory on its way into the Nepali hills, adding a layer of shared migratory history between the two countries that a simple north-south genetic divide would never reveal. The Sherpa case adds a second layer of complexity to this comparison, one that touches on timing rather than direction. Popular understanding generally treats the Sherpa as one of the Himalayas' most ancient peoples, their presence in these mountains taken for granted as a permanent ancient fixture of the landscape. Genetic research tells a considerably more recent story. A detailed study analyzing DNA from more than 500 Sherpa individuals living across both Nepal and Tibet found that Sherpas share the overwhelming majority of both their paternal and maternal genetic lineages directly with indigenous Tibetans, representing what researchers describe as a recently derived sublineage rather than an ancient, independently established Himalayan population. Using genetic dating techniques applied to specific Sherpa mitochondrial sub-haplogroups, researchers calculated that the genetic divergence between Sherpa and broader Tibetan populations occurred less than 1,500 years ago, meaning the Sherpa people's presence in Nepal represents a comparatively recent migration event, considerably more recent than the ancient multi-thousand-year timelines most people instinctively associate with Himalayan settlement, and considerably more recent than the deep multi-thousand-year timeline connecting India's own ancestral North Indian and ancestral South Indian populations, which trace back tens of thousands of years on one side and several thousand years on the other. This timing difference matters directly for understanding the India-Nepal genetic relationship as a whole. India's own population history, as this channel has explored extensively, reflects multiple ancient migratory waves stretching back tens of thousands of years, layered with a documented Bronze Age steppe migration arriving roughly 4,000 years ago. Nepal's population history, by contrast, includes at least one genuinely recent migratory event, the Sherpa settlement of the High Everest region, occurring within the last 1,500 years, layered on top of considerably older Tibeto-Burman and Indo-Aryan ancestral threads. The two countries' populations weren't simply formed by the same ancient process playing out on both sides of a modern border. Nepal's specific ethnic mosaic includes chapters of population history, like the Sherpa migration, that have no direct parallel anywhere in India's own genetic story. Formal statistical analysis of gene flow direction adds further precision to this comparison. Using a technique specifically designed to detect the direction of ancient population movement, researchers found considerably stronger evidence for gene flow moving from Tibet southward into Nepal than for any significant reciprocal movement of Nepalese populations northward into Tibet. No equivalent large-scale statistical study has specifically measured the direction of gene flow between Nepal's Terai populations and India, but the near-total genetic overlap between Nepali Chetri and Brahmin communities and their Indian counterparts suggests a relationship considerably more like a single continuous population than the more clearly one-directional Tibet to Nepal flow documented for the country's hill regions. This genetic finding connects directly to a real, documented modern political and social reality that has shaped Nepal-India relations for generations. The 1950 Treaty of Peace and Friendship between the two nations formally established the open border and reciprocal citizenship rights that still govern movement between them today. A policy framework that this genetic evidence now confirms was built on top of a population relationship already centuries, if not millennia, in the making. Nepal's Madhesi community concentrated specifically in the Terai Plains, and carrying the same close genetic connection to northern India, has long occupied a complicated position within Nepali national politics, sometimes facing discrimination and questions about their authentic Nepali identity from hill region political elites, despite representing one of the country's largest population groups. The genetic evidence explored throughout this documentary doesn't resolve that specific, sensitive domestic political question, which involves citizenship rights, political representation, and cultural recognition extending well beyond population genetics. But it does confirm with real scientific precision that the ancestral connection Madhesi communities maintain to northern India isn't a recent or superficial one. It reflects a genuine, deep, and remarkably well-preserved population relationship that predates the modern Nepali state itself by a considerable margin. There is a caste and community dimension to this comparison worth examining as well, because it reveals something the simple geographic story misses entirely. Genetic researchers note that Brahmin communities specifically are commonly classified within Nepal's Indo-Aryan lineage, despite frequently living in the upper hill regions, geographically embedded among predominantly Tibeto-Burman neighboring communities, a reminder that in both Nepal and India, caste and community-based marriage practices have historically mattered more in shaping genetic ancestry than simple geographic proximity. A Nepali Brahmin family living in the hills, surrounded by Tamang or Rai neighbors, maintains a genetic profile connecting it to India and the broader Indo-Aryan world, not to the geographically closer Tibetan-related communities living in the very same hillside villages. This same pattern, caste and community mattering more than geography, appears consistently across India's own internal population structure, confirming that the same underlying social forces shaping genetic diversity within India also operate identically across the open border in Nepal. Genetic analyses measuring runs of homozygosity, long stretches of genetically identical DNA that increase in frequency when a population has historically married within a relatively small, closed community over many generations, found elevated levels across Nepal's Indo-Aryan, Magar, Newar, Rai, and Tamang populations, alongside specific Sherpa communities. This finding mirrors the broader pattern of persistent endogamy documented across India, where more than 4,000 distinct endogamous communities have preserved distinct genetic signatures for centuries, despite living within the same broader national population. Nepal, in this specific respect, functions as a genuine extension of the same social and genetic pattern found throughout India, rather than an entirely separate demographic system. This shared pattern of community-based marriage carries the same practical medical significance in Nepal that this channel has documented extensively across India and Pakistan. Elevated endogamy rates, whether in a Kathmandu Newar family, a hill region Tamang community, or a Terai Brahmin household, tend to concentrate specific rare recessive genetic conditions within particular populations, a pattern requiring exactly the kind of population-specific medical genetics research India's own Genome India project and Pakistan's Genome Resource have prioritized in recent years. Nepal's own comparatively smaller genomic research infrastructure means the country currently has less detailed population-specific genetic reference data available for its many distinct ethnic communities than either of its larger neighbors, an ongoing gap that researchers studying South Asian genetic diversity have flagged as a priority for future regional research collaboration. None of this diminishes the reality of a shared modern Nepali national identity, forged through the unification of dozens of previously independent hill kingdoms and valley civilizations under the Shah dynasty beginning in the 18th century, or the reality of India's own distinct national identity and history. Nepal and India each represent genuine, meaningful political and cultural achievements built by real communities across real centuries of shared history. Genetic ancestry doesn't compete with or diminish either nation's identity. What the comparison reveals is that the relationship between these two neighboring populations is considerably more precise and considerably more variable by region and community than the simple story of two friendly neighbors sharing a border and a broadly similar culture would suggest. For scientists, that is the real result of comparing DNA across India and Nepal. Not a single uniform answer describing how closely related the two nations' populations are, but a genuinely precise, region by region and community by community picture. Nepali Terai populations essentially indistinguishable from northern Indian populations across the open border. Nepali hill and mountain populations connected overwhelmingly to Tibet, sharing almost nothing genetically with India. The Newar of Kathmandu sitting at a genuine, historically earned midpoint between both worlds, and at least one specific community, the Sherpa, carrying a genetic history so recent and so specifically tied to Tibet that it has no real parallel anywhere in India's own considerably older population story. There's a final comparison worth drawing before this story closes, one that places the India-Nepal relationship in useful context against the other cross-border genetic comparisons this channel has explored in detail. Punjabi communities across the India-Pakistan border remain genetically nearly indistinguishable despite 80 years of hostile political separation and multiple wars. Sindhi communities show 60 to 66% shared ancestry across that same militarized frontier. Balak populations across the Iran-Pakistan border show genetic distance close to statistical zero. In every one of these cases, deep genetic continuity persisted, despite political division, conflict, and in some cases active hostility between the nations involved. India and Nepal offer a genuinely different kind of case study. Two nations with no history of war between them, an open border, and deliberately close diplomatic ties. And the genetic evidence confirms exactly what that friendly relationship would predict for the specific populations living closest to the shared frontier, while simultaneously revealing that friendship and open borders alone don't erase the deeper, older ancestral distinctions separating Nepal's hill and mountain communities from India entirely. Political relationships, whether hostile or friendly, consistently turn out to matter less to the underlying genetic story than the much older, much more patient forces of geography, migration, and marriage that shaped these populations over thousands of years. That is what scientists found when they finally compared DNA across India and Nepal. Not a simple blend zone gradually transitioning from Indian to Tibetan ancestry as you travel north, but a genuinely precise mosaic, where an open border allows one specific set of communities to remain essentially one continuous population with northern India. While, just a short distance further into the same small country, an entirely different set of communities carries an ancestral story connecting them instead to Lhasa, to the high Tibetan plateau, and to a migration event barely 1,500 years old, proving that even between two of the friendliest, most closely connected neighboring nations anywhere in Asia, the genetic map still refuses to follow the same simple lines drawn across the political one.